created by MindSticks Foundation Trust.
Welcome, students and learners. Today, we are taking a truly fascinating journey in this explainer. We will start by understanding the foundational mathematics of lenses, magnification, and lens power. Then, we will explore the biological marvel that is the anatomy of the human eye, followed by vision defects and their corrections. After that, we'll see how we use powerful optical instruments to look at the very small and the very far. And finally, we will explore exactly how we perceive vision inside the brain. It's a beautifully structured path, so let's begin.
Okay, let's dive into this. Section 1, Magnification and Lens Power.
So magnification is essentially a ratio. You're just comparing the height of the image to the height of the original object, or the image distance compared to the object distance. Pretty straightforward, right? But what about power? Well, power represents a lens's capacity to either converge or diverge incoming light rays. It is simply the inverse of the focal length in meters, and we measure it in a unit called diopters. Keep these principles in mind because they are the absolute foundation of everything we're going to discuss today.
Now, what happens if you combine two lenses together? The math is actually beautifully simple. When two lenses are kept touching each other, calculating their total effective power is as easy as adding their individual powers together. So P equals P1 plus P2. This incredibly simple additive property is exactly how we build complex optical systems like microscopes.
Let's move on and see how this builds. Section 2. Anatomy of the human eye.
Think about it. Light first enters your eye through a thin, transparent cover called the cornea. Then the pupil, that dark opening right in the center of the iris, acts like a gatekeeper, controlling exactly how much light enters. Just behind the pupil is the crystalline lens. This lens refracts the light to focus a real inverted image onto a very delicate screen of light-sensitive cells at the back of the eye. We call this screen the retina. It is quite literally a perfect optical machine.
But here's the catch. Our eye isn't just a rigid piece of glass. It possesses something truly remarkable called the power of accommodation. Notice those ciliary muscles holding the lens? These muscles physically change the shape of the crystalline lens. When you look at distant objects, the muscles relax and the lens becomes flat. But when you look at something nearby, the muscles contract, the lens becomes much more rounded, and its focal length actually decreases. This physical movement is exactly how we constantly keep the world in sharp focus.
Of course, for a normal, healthy eye, there are clear limits to this vision. We can clearly see objects as close as 25 centimeters away from our face. This is known as the minimum distance of distinct vision, or simply the near point. And the farthest distance, the far point? Well, it is at infinity. A healthy eye can span that entire immense range without experiencing any stress whatsoever.
But sometimes things don't work perfectly. Section 3. Vision defects and their corrections.
And this brilliantly illustrates what happens when refraction goes wrong. In nearsightedness or myopia, the eyeball elongates or the lens becomes too curved, causing the image to form completely in front of the retina. On the flip side, we have farsightedness or hypermetropia. Here, the eyeball is flattened, meaning the image forms way behind the retina. In both of these cases, the final result for the person is unfortunately a very blurry picture.
But this is exactly where we use the physics of lenses to solve our biology. To correct myopia, we prescribe spectacles with a concave lens, which has a negative power. It diverges the light rays just enough before they enter the eye so they land perfectly on the retina. For hypermetropia, it's the opposite. We use a convex lens with positive power to converge the rays a bit earlier. Problem solved.
Now, there is also a very common condition called presbyopia. You see, as people age, their ciliary muscles generally weaken and the lens loses its ability to change shape so easily. So, the near point shifts farther away. Sometimes, older people actually suffer from both nearsightedness and farsightedness at the exact same time. To correct this, they require bifocal lenses, where the upper part is a concave lens for viewing distant objects and the lower part is a convex lens to help them read.
Moving beyond the eye itself. Section 4. Powerful optical instruments.
You know, these lenses are actively working all around you every single day. We use concave lenses in medical scanners, the peepholes in our doors to see a wider area, and even inside torches to spread out the light produced by the bulb. Meanwhile, convex lenses are heavily used in simple microscopes, cameras, and the spectacles we just discussed. So the science you are learning today is constantly at work in your daily life.
Take the microscope, for example. A simple microscope uses a single convex lens with a small focal length to magnify objects about 20 times.
which is absolutely great for things like watch repair. But a compound microscope uses two lenses, an objective lens and an eyepiece. By combining them, just as we learned in our very first section, we get a much higher two-stage magnification. This is what allows us to study minute living things like bacteria and blood cells.
Now contrast the microscope, which looks at the tiny, with the refracting telescope, which looks outward to the vast stars. A refracting telescope uses a very large objective lens with a much larger focal length. Why? To collect the maximum amount of light possible from distant astronomical sources. The smaller eyepiece lens then forms the final magnified image for the astronomer to see.
Finally, let's explore the brain's role. Section 5. How we perceive vision.
So the crucial point here is a phenomenon called persistence of vision. When an object is suddenly removed from your sight, the image doesn't instantly vanish from your retina. No, the sensation actually persists, imprinted right there for 1/16th of a second. This tiny delay is vital. It is exactly what allows us to perceive continuous reality, blurring rapidly changing static images into smooth, flowing motion.
Now, what's really interesting about our retina is its cellular makeup. It is made of two main types of light-sensitive cells. Rod cells respond to the intensity of light, giving our brain information about the brightness or dimness of an object. Conical cells, on the other hand, respond to color. They are the entire reason we can perceive the beautiful reds, greens, and blues of our world.
But what happens when this biological system isn't perfectly tuned? Well, this leads to color blindness. This condition simply means that specific conical cells are unresponsive to certain colors. They just do not get excited by red or green light, for instance, and fail to send those specific signals to the optic nerve. It is very important to note, though, that this doesn't affect a person's general everyday eyesight. They just cannot distinguish between certain specific colors.
Which brings us to a final profound question to end our explainer today. Considering the persistence of vision and how our specific rod and conical cells individually dictate what we see, just as we observe in color blindness, do we all actually see the exact same reality? Or is the universe we experience every single day just a construction of our own personal lenses and retinas? Think deeply about that the next time you look at the vibrant world around you. Thank you for learning with me today.